Hello students, welcome back to the second part of Cognitive Neuroscience lecture video. In this video, we'll be continuing our discussion in the topic of cognitive neuroscience, and particularly about localized representation. So one of the basic principles of brain organization is localization of function. So the specific functions are served by specific areas of the brain, as you may already know about this. And many cognitive functions are served by the cerebral cortex, which is a layer of tissues about 3 millimeters thick that cover our brain. Other functions are served by subcortical areas that located below the cortex. And early evidence for localization of function came from neuropsychology, which is the study of the behavior of people with brain damage. So in the early 1800s, an accepted principle of brain function was cortical equipotentially, and the idea that the brain was operated as an indivisible whole as opposed to specialized area. But then in 1861, Paul Broca published work based on his study of patients who had suffered brain damage due to strokes that caused disruption of the blood supply to the brain. So these strokes actually caused damage to an area in the frontal lobe that came to be called Broca's area, as you can see in this figure. So, and then after that, 18 years after Broca reported on his frontal lobe patient, Carl Wernicke in 1879 described a number of patients who had damage to an area in their temporal lobe that came to be called Wernicke's area. As you can see in this figure, here is Wernicke's area. And then Wernicke's patient produced a speech that was fluent and grammatically correct, but tended to be incoherent. So here is a modern example of the speech of patient with Wernicke's aphasia. Yeah, patients such as this not only produce meaningless speech, but are unable to understand other people's speech, even though they're able to talk. Yeah. So their primary problem is their inability to match words with their meanings, with the defining characteristic of Wernicke's aphasia being the absence of normal grammar. So Broca's and Wernicke's observations show that different aspects of language, the production of language and comprehension of language, were served by different areas in the brain. And as we will see later in this video, modern research has shown that the strict separation of language function in different areas was an oversimplification, actually. Nonetheless, Broca's and Wernicke's 19th century observation set the stage for later research that confirmed the idea of localization of function. And several research in neuropsychology also found that damage in occipital lobe or the visual cortex caused blindness, and damage in the left occipital lobe caused blindness to the upper right side, and damage in down right side caused prosopagnosis, or could not recognize faces, and damage in temporal lobe caused problem in hearing, damage to parietal lobe caused problem in touching, pressure, and pain, and damage to the frontal lobe affected the all sensation coordination, thinking, and problem solving. And another tool for demonstrating localization of function is recording from single neurons. So what happened? So numerous studies, mostly on animals, used a single neuron recording to demonstrate the localization of function. For example, an experiment by Doris Sau and co-workers found that 97% of neurons within a small area in the lower part of monkey's temporal lobe responded to pictures of faces, but not pictures of other types of objects. So that's why this face area, as it turns out, is located near the area in humans that is associated with prosopagnosia. So the idea that our perception of face is associated with a specific area of the brain is also supported by research using the technique of brain imaging, which make it possible to determine which areas of the brain of humans are activated by different cognition. As we noted in chapter 1, that technological advances that causes a shift in the way science is done can be called as revolution, moreover in cognitive psychology. And on that basis, it could be argued that the introduction of the brain scanning techniques, the PET or the positron emission tomography, and the fMRI or functional magnetic resonance imaging marked the beginning of the imaging revolution. So brain scanning and especially fMRI has played an important role in understanding the physiology basis of cognition. So here we consider what fMRI research tell us about the localization of function in the brain. And we begin by describing the basic principle behind fMRI. So many of the brain imaging experiments that have provided evidence for localization of function have involved determining which brain's areas were activated when people observe pictures of different objects. So basically, fMRI here sees the oxygen level in the blood. So the tools can record the oxygen flow. Therefore, we can see which part of the brain are activated. That we can see in this picture is the foxel. So the colors that the colors in the record convey the brain activity, which is the red and yellow, are increasing, while the blue and the green means the activity is decreasing. All right. So localization is demonstrated by brain imaging. So the example is the research conducted by researchers that investigating when a person looking at pictures, and they found out that the fusiform face area or FFA is active when looking at picture of faces. But the parahippocampal place area or the PPA is active when perceiving indoor or outdoor scenes, while the EVA or the extrastriate body area is activated by pictures of bodies and part of bodies, but not faces. And then another example of research by Huss. So the participants were asked to looking at the movie. So seeing scenes that contain different moving objects. Then who here analyzes how the foxels in the participant's brain responds to different object and action in the films. Yeah. So the noun and verb appear in the categories or the labels from the fourth scene here. And from whose experiments by brain imaging shows types of stimuli that causes foxels across the surface of the brain response. So as you can see in this figure, the area labeled humans at the bottom of the brain here, which is actually on the underside of the brain. Yeah, actually this is underside of the brain, corresponds to the fusiform face area, which responds to all aspects of the face. So the area labeled humans higher on the brain here responds to specifically to facial expression. And the areas labeled as talking here corresponds to Broca's and Wernicke's ideas. So by showing the location on the brain where by the indicated categories are most likely to activate this different part of the brain area. And objects and action here similar to each other are located near each other in the brain. So so let's consider Hood's maps of the category in the brain in the previous figure that shows that there are two two location of humans categories, right? And the explanation of that is the different areas respond to different features of humans. It is illustrate a central principle of cognition actually. So most of our experience is multi-dimensional. That is, even simple experiences involve combination of different qualities. So consider, for example, looking at a person's face. So the idea of distributed representation is that the specific functions are processed by many different areas in the brain, not only one part. And looking at a face activates many areas of the brain is called as distributed representation. So for example, the fusiform face area or the fusiform gyrus that we talked earlier, it is strongly activated by faces and respond more weakly to other types of stimuli. This is right. But just because there is an area that is specialized to respond to faces doesn't mean that face is activated only in that area, only in the fusiform face area or gyrus. So when you see someone's face, brain also respond to other various information to a face. For example, the emotion. All right. And distributed representation also we can observe in other cognitive functions such as remembering. So remembering contains memories that are actually complicated. And from the brain imaging, it shows area activated by episodic memories shown in yellow areas and the semantic memories shown as the blue areas. So episodic memories are memories for events in a person's life, like remembering what you did yesterday. And semantic memories are memories for facts, like knowing the capital of Spain is Madrid. So this is like the same cognitive function, the memory, but then the representation in our brain are distributed, not only in a single area, as well as producing and understanding language. So the pathways shown in blue lines here involve in processing sound, producing the speech, and saying the words. But the pathways showing in the red line are involved in understanding the words itself, with both set of pathways being involved in understanding sentences. So again, the distributed representation of producing and understanding language is distributed in a lot of areas of the brain and not only single areas like Broca's or Wernicke's area. And neural networks, we're discussing about neural networks then, it actually are interconnected areas of the brain that can communicate with each other, right? And the idea of neural network is a logical extension of the idea of distributed processing, because it makes sense that if many areas are involved in a particular type of cognition, and that they might be connected. They're talking to each other, they have communication, right? As we tell the story of how then researchers are discovering the properties of neural networks, we will be introducing this 4 principles. So the first one is there are complex structural pathways called network that form the brain's information highway, right? So the complex structural pathway. And the second one, within this structural pathways, there are functional pathways that serve different functions. And then the third one, these networks operate dynamically, mirroring the dynamic nature of cognition. Okay, so the pathway is not only like a single line, but then it can be like a complicated line, okay? And the fourth one, there is a resting state of brain activity. So parts of the brain are active all the time, even there is no cognitive activity. So we begin by considering how neural network has been described structurally, right? And different experience will be also represented in the brain through the activation of different neuron patterns. And as you can see in this video, this is the example of pain matrix neural networks that consist of the communication or the activation of several parts of the brain, including thalamus, dorsolateral prefrontal cortex, anterior cingulate cortex, and so on. And then, so determining the brain's wiring diagram is an important step in understanding how different areas of the brain communicate, yeah, because communication depends on structural connection. So, so by knowing the brain's wiring diagram, it will that is created by nerve axon that connect different brain's areas, right? And then researchers determine this connection by using classical neuroanatomical techniques, and which slices the brain tissue were stained to highlight the axon by the microscope. It was in the past, but now a new technique called TWI, or track weighted imaging, which is based on the detection of how water diffuses along the length of nerve fiber. So we can understand the structural connection in such a cognitive function inside our brain. As you can see in this figure, the TWI produce a very beautiful structural connection of our brains. And then different parts of the brain's neural networks are actually involved in carrying out different cognitive or motor tasks. But then how is it possible for us to determine what parts of the neural network are involved in different connection, or sorry, the different function, right? So one way this question has been answered is by measuring the functional cognitivity, yeah? So in the previous slide, we're talking about structural connectivity, so which part of the area of the brain, but now we're talking about functional connectivity. With functional connectivity being determined by the extent to which neural activity in two brains area, the left side and the right side, are correlated. So if the responses of two brain areas are correlated with each other, this means that they are functionally connected, right? So one method of determining whether the responding of two areas is correlated is based on the resting state fMRI. Yeah, is the fMRI response measured while a person is at rest, that is not performing any cognitive task. And then functional connectivity here being determined by the extent to which neural activity in two brain areas are correlated. So if the response of the two brain areas are correlated with each other, this means that they are functionally connected. So as you can see in this figure, so left hemisphere of the brain showing the seat location of motor with the symbol L. In the left motor cortex, yeah, and then and a number of task locations, each indicated by the dot here, the black dot. And the task location motor R is in the right motor cortex here, yeah, and it's in the right motor cortex on the other side of the brain from motor L, yeah, and the the task location somatosensory is in the somatosensory cortex here, which is involved in perceiving touch. So by using resting state fMRI, the resting level fMRI response of the motor L is also called seat location. And then the resting level fMRI response of the somatosensory test location here is the showing in the C diagram here. So the response in B and C are for second long. Okay, so from this research using the resting state fMRI, we can see the functional connectivity between the motor L and the somatosensory function of the brain. And the number of correlation then calculated by a complex math procedure that compares the seed and the test response along the horizontal time axis. Yeah, so when it has high correlation means that it indicates high functional connectivity. So the other location other than the motor and somatosensory have low correlation, so are not functionally connected to the seed location. And then a resting state functional fMRI connectivity then has become one of the main methods for determining functional connectivity of our brain. And this figure shows the network that is determined for a number of different functions using these procedures. As you can see, the 6 common functional network and its location in the brain, you can see it from this picture. And the previous picture also show that the overall structural map of the brain is divided into smaller functional map, right? So different cognition activate different groups of neurons, as you can see in the figure. By the color of red and yellow, it means that the neural is firing, right? But to really understand what is happening during the cognition, we need to go beyond just identifying areas that serve different functions. We need to consider the dynamic of the cognition. So explanation here that everyday experience like looking at and picking up a cup of coffee involve rapid switching and sharing of information between a number of different functional networks, right? So what is happening when a person looks at a cup of coffee on the table? So looking at the cup causes activity in the visual function network, right? But then as the person perceives the various quality of the cup, then it activated the network of visual function in different way. Meanwhile, the attention network may also be activated as the person focuses attention on the cup, and then the motor network becomes activated as the person reaches to pick up the cup, grabs it, and leave it to drink. So here's to illustrate how dynamic is our cognition that requires different neural networks in our brain. So by doing a simple task like drinking the cup, but it activating a lot of neural network in our brain. And so our brain might switch from task-related networks involved in driving to our default mode and network, or DMN. So as you might expect, mind wandering during driving, yeah, we usually do this, we often do this, as well as during other tasks besides driving, isn't necessarily a good thing for performance. So this idea is confirmed by a large body of research that shows that mind wandering decreases performance on tasks that require focused attention. So so when we're mind wandering, we kind of like activating this default mode and network, yeah. And it is not only about decreasing performance on tasks by distracting you from your works, but it's also involved in processes ranging from attention to memory to creativity as well. And large body of research still been done, still been doing to explain more about default mode network. So in this figure, so this is the brain areas that decrease their activity during the task performance because of the default mode and network, yeah. And this figure or this diagram is the resting state activity at 2 points in the right hemisphere, indicated by the arrow that above. The fact that the resting state activity is correlated indicates that these areas are functionally connected, actually. All of these areas taken together are called the default mode network. All right, so we covered a lot of ground in this chapter, all the way from early neuropsychological research by Broca and Wernicke on how brain damage affect language, to how single neuron respond to visual stimuli, to how multidimensional cognitions are represented by the dynamic activity in the interconnected neural networks. So thank you for your attention in this video, and I will see you again in our next classes. Take care and thank you.